Hi, and welcome. I am Amy Myers. Welcome to my presentation on the article, Small CRISPR RNA's Guide Antiviral Defense in Prokaryotes, authored by Stan J.J. Brahms et al. This article appears in the August 15th, 2008 issue of the journal Science and is a concise account of the author's research as they explore CRISPR and its primary function. As a note, and I'm going to say this throughout the entire presentation, given the age of this article in the time after its publication, we have learned so much more about CRISPR and its function. But it's important that we explore the history of CRISPR research. Brown's article provides an excellent example of using data from one experiment to inform study choices in the next. In other words, the authors followed the clues in order to better understand CRISPR and its function. Throughout this presentation, please bear in mind that this is one of the first studies that actually grasps CRISPR for its actual mechanism and function. This was a huge step in CRISPR research. The purpose of this article, according to the authors, is to demonstrate several possibilities related to CRISPR and their associated proteins in regards to their function in immune response against viruses. First, the authors wish to demonstrate how virus-derived sequences found in CRISPRs are used by CRISPR-associated or Cas proteins. The authors seek to establish how they together mediate an antiviral response that protects from infection. Secondly, the authors wish to describe how post-CRISPR transcription, a complex known as cascade, cleaves a CRISPR RNA precursor in each repeat, keeping the cleavage products which contain the virus-derived sequence. Next, the authors explore how these mature CRISPR RNAs function as small guide RNAs that allow CRISPR to interfere with virus reproduction. Finally, the authors plan to demonstrate that the formation of mature guide RNAs by the CRISPR RNA endonuclease, subunit of cascade, is a mechanistic requirement for antiviral defense. Those are four fairly lofty goals, and the authors are going to take us through these really in a very systematic manner. But now that we know what they want to convey in their article, let's take a moment to review a few terms and concepts that will help us delve a bit deeper into this material. What is CRISPR? It was not officially discovered until 1987, when it was cloned by accident. But even then, it was just recognized as a pattern found in E. coli. It would take more than 10 years for researchers to note how prolific that special pattern of short repeat, spacer, short repeat was. CRISPR stands for clustered, regularly interspaced short palindromic repeat, which is a reference to its most defining characteristic, that pattern. Francisco Mojico created a plasmid containing a CRISPR fragment in 1995. When used, it altered the distribution of the Haliopteryx vulcanii genome. This is an archaea. It would be longer still before scientists recognized that CRISPR was responsible for a form of immune defense in bacteria and archaea. Specifically, the discovery of the Cas genes, or CRISPR-associated proteins, allowed researchers to grasp the unique qualities of CRISPR in acquired immunity. Part of that immunity included the concept of precursor CRISPR RNA, or pre-CR-RNA. In 2012, 2 labs recognized that the CRISPR-Cas system together was capable of cutting DNA. This is when the possibility of utilizing CRISPR-Cas for gene editing became a reality. By 2013, CRISPR-Cas9 had successfully edited functional genes in both humans and in mouse cells. Overall, CRISPR, along with Cas genes, function as a type of acquired immunity to bacteriophages for both bacteria and archaea. And as we will see, it also has a role in other organisms as well, including a similar system in eukaryotes. RNA interference, or RNAi, small interfering RNA, or siRNA, and RNA-induced silencing complex, which is referred to as RISC complex, are all part of the eukaryotic system. CRISPR has some absolutely remarkable characteristics. CRISPRs themselves vary in length and sequence of their repeated sections in such a way that they can be categorized based on sequence similarity using phylogenetic principles. And considering it's based on what the organism's evolutionary lineage has been exposed to, it makes sense. Some are palindromes and produce RNA molecules that are stable with hairpin structures, but most CRISPR repeats have a conserved repeat of G, A, and then either C or G motif, which is found at the 3' end, and may allow for Cas proteins to bind. CRISPR loci are often flanked by adenine and thymine-filled sequences that we refer to as leaders. The spacer closest to that leader tends to be the most diverse, while those further away tend to degenerate. This indicates that the sequence has polarity, and we now understand that the leader sequence holds the promoter elements and the binding sites for those regulatory proteins. Overall, prokaryotes are going to respond to changes in their environment by utilizing signaling cascades, many of which lead to transcriptional reprogramming. Cyclic AMP, otherwise known as CAMP, controls some of the Cas genes and seems to serve as a signaling molecule to stimulate the immune system by binding to and activating transcription factors. CRISPR is not the only immune response, but the specific CRISPR system activated tends to be in response to the pathogen attacking. As you can see in Figure 1A, which was found in a different article created by Sorek, Lawrence, and Weidenheft in 2013, it explains in a simple form the manner in which CRISPR functions to help protect bacteria and archaea from phages. Here, you can see the 3 phases of CRISPR's basic function: CRISPR adaptation, CR-RNA biogenesis, and CR-RNA-guided interference. That first phase of adaptation or acquisition occurs when CRISPR loci incorporate homologous sequences from the challenging phage into their spacer sequence. This process happens relatively quickly, giving the bacteria an advantage over the specific pathogens. These spacers are typically added near the leader end of the CRISPR and are accompanied by an additional repeat section. That trailer end of the CRISPR loci, where we previously mentioned degeneration tends to occur, serves the purpose of deleting outdated spacers. In this way, quite brilliantly, the genome can remain compact and it remains relevant. That second stage, or biogenesis, we also refer to it as expression and processing phase, is when the entire CRISPR array is transcribed as a long precursor RNA, which we also refer to as pre-CR-RNA. The all-important Cas proteins then process this precursor into short, mature CRISPR RNAs or CRRNAs. Each CRRNA contains one spacer sequence and functions as guide RNAs for the defense phase, which is our third phase. The interference or defense phase is when the CRRNAs associated with Cas proteins form a large molecular complex, which is referred to as a cascade or CRRNP. This complex is guided by the CRRNA's spacer sequence to the complementary sequence on the invading nucleic acid, leading to the invader's destruction or obstruction to prevent it from replicating. For many well-characterized systems, the target is the DNA of the invading phage or plasmid. This is a major distinction from the eukaryotic RNA interference, RNAi, which typically targets just RNA. In addition, this system avoids attacking its own CRISPR DNA because the CRRNA uses a small segment of the repeat sequence at the 5' end for base pairing. The complementarity that exists only with the host's own CRISPR DNA effectively allows interference only against targets that lack this specific sequence. So without that sequence, the DNA is fair game for a CRISPR attack. Just for additional information, that previously mentioned RNAi system found in eukaryotes is a similar system for targeted immune response. Both systems do rely on generating small guide RNAs. For prokaryotes, it's the CRRNAs. For eukaryotes, it's actually that siRNA from a larger precursor and using a protein complex. The protein complex in prokaryotes is the Cas complex, and in eukaryotes, it is known as the RISC complex, RISC. To target, and these complexes, of course, do the same thing. They target and cleave foreign nucleic acid. While we're talking about the main function of CRISPR, researchers in the last decade have found numerous uses for CRISPR and its associated proteins. The medical applications are nearly endless at this point, including identifying DNA segments, editing genomes, and use in testing. The field of biosensing, however, has recently exploded in opportunities. Electrical transduction allows technologies such as resistive pulse and conductivity sensing, while optical transduction allows us to use things like luminescence, fluorescence, and gel electrophoresis in biosensing as well. And these are just some of the more recent applications. Use of CRISPR to build better crops, genetically modified organisms, and improve consumer products really took off in 2021. And of course, while these were not the first times that these particular things had been done or created, this is the first time, 2021, when we start to see it commercially. As we move into a more thorough discussion of this article, again, keeping in mind it was written nearly 2 decades ago, well before these applications were even a twinkle in a researcher's eye. We have come so far in CRISPR technology, but as with all things, understanding how we got here is imperative to seeing where we can possibly go next. So, I am going to give a quick rundown on some of the specific methods used. I'm going to talk about some of the chemicals, some of the kits, and some of the strains that were utilized. These were not actually all included in the original article provided. I was able to track down the author's supplemental material through the journal Science. In the supplement, the authors not only included the specifics of their methodology, but they also provided a more in-depth look at the eukaryotic system that is comparable. Later in this presentation, I am going to go through the experiments in a more explanatory manner, but for now, feel free to scan this QR code, log in using your WKU credentials, and you should be able to read along through the methods section. You'll be able to see the things that they chose not to explicitly state in the article. All right, let's talk strains. There are a couple of different bacterial strains that are going to be mentioned by the authors, but they really highlighted this main strain of Escherichia coli, K12. They specifically used the gene knockouts from the KEIO collection. During the gene cloning, protein production, and purification, Cas genes and CRISPRs were PCR amplified from the K12 E. coli strain known as W3110, which is just an indicator of the genomic DNA the E. coli strain carried. It was directionally cloned to a compatible expression vector set. Primers for the Cas genes were created based on predicted translation start sites. Then they would use the quick change site-directed mutagenesis kit, and it was used, of course, per its description, to induce mutations. 0.1 millimolar IPTG was used to induce protein production. Then cells were harvested and resuspended in 20 millimolar tris HCl supplemented with a 0.1 sodium chloride and disrupted using the French pressure cell. Tagged proteins were isolated using streptactin, his select or amylose affinity chromatography, the specifics of which were predicted by the use of mass spectrometry. Moving on to the methods used in northern blotting. Total RNA was collected from 11 millimeters of exponentially grown E. coli and was isolated using the Mirvana miRNA isolation kit. And with any of these kits mentioned, the authors are using the standard protocols. E. coli strain BL21 with various plasmid combinations did not have IPTG induction, which did give them low Cas gene expression. Northern blots were run with 10 micrograms of RNA on a 9% polyacrylamide gel using 7 molar urea in a 0.5 times TBE buffer. This would later be used to analyze the products as well. Blots were prepared, and they described the process, which was a very complicated and involved process. But then they were left for 12 hours to be probed. They were probed with a P5' labeled oligonucleotide of spacer 4. Once that step was complete, that actually brought the results from that brought the authors to the cleavage reactions using internally radio-labeled transcripts that were created by in vitro transcription using the MaxiScript T7 kit, again with standard protocols. Templates for in vitro transcription were generated by polymerase chain reaction, PCR, and full-length RNA substrates were isolated by gel from denaturing a 2% agarose. RNA cloning was used, and they did that, they accomplished that by using protein-bound total RNA, which was isolated from that streptactin purified cascade that was isolated using the Mirvana miRNA isolation kit, standard protocols. 6 micrograms of RNA was denatured for 10 minutes at 65 degrees Celsius and subsequently 3' polyadenylated for 75 minutes at 41 degrees Celsius using poly-A polymerase. RNA products were separated from unincorporated nucleotides and enzymes using a Nucleaway gel filtration spin column and reverse transcribed with the Superscript 3 kit for 15 minutes at 50 degrees Celsius with anchored primer BG2164. The RNA strand was then degraded. Single-stranded DNA was purified, and a 3' tail was added during the 60-minute incubation that followed. Moving into the phage studies. These studies used a virulent variant of phage lambda and E. coli BL21-AI strain as a host. The strains were grown in lambda media, and cells were prepared and used on plaque assays that were completed in triplicate. At this point, I would really like to dive a little bit deeper into what the authors were trying to accomplish using these methods. As a 2008 paper, again, I told you I'm going to harp on this a little bit, the authors are attempting to understand the mechanism by which the CRISPR-Cas system functions. To begin, the researchers isolate and produce the endonucleases in question. They used E. coli strain K12, again, to explore 8 specific Cas genes. So these Cas genes were Cas3, a set of 5 genes together referred to as Cas A, B, C, D, E, Cas1, and Cas2. These genes were tagged at the N and C terminus, then affinity purified. The tagging did allow for identification of the proteins in question. As you can see in this schematic, denoted as Figure 1A, the repeats are going to be indicated here by diamonds, while the spacers are shown with rectangles. If you will look at the section that sits at the top of the schematic, you can locate the convergently pointing arrows along that sequence. That's where the palindrome is located. The complex of 5 proteins, that Cas A, B, C, D, E, is actually referred to as the cascade, or CRISPR-associated complex for antiviral defense, which I felt was an incredibly clever name. Using protein electrophoresis with Kumasi blue stained SDS polyacrylamide gel, the affinity purified protein complex can be seen in Figure 1B. Each bar, of course, indicates a collection of proteins that were tagged at the N terminal with strep2 tag or at the C terminal with his tag. All subunits were utilized as bait to capture the appropriate proteins. As we go through this presentation and you start to see how this CRISPR is working, or based on some of the review articles that you've read, you can see why bait is actually a really appropriate term, because those segments are what are matching up to the viral DNA and causing the phage DNA to be attacked. The authors note that Cas A was not a successful bait. The asterisks do indicate where the double tag subunits landed, and this is important because due to the double tagging process, They're actually 5.5 kilodaltons larger and therefore traveled to a different section of the gel. On the left, the authors have included the marker in kilodaltons, and on the right, they did add labels for the untagged subunits. The next step along the path of discovery is the exploration of the cascade function. For this, the authors analyzed the effect of in-frame Cas gene knockouts on the formation of transcripts in the CRISPR region. Using a northern analysis of total RNA, as seen in Figure 2b on the right, the authors used a single-stranded spacer sequence as probes to show that transcription was taking place in the downstream regions of the Cas2 gene, but it's not taking place in the other direction. Looking at the same figures, next we're going to talk about the control strains, including wild type and non-Cas gene knockout strains. From this exploration, they discovered that a small CRRNA of around 57 nucleotides, and this 57 nucleotide segment of RNA is actually going to become incredibly important to the author's understanding of how CRISPR is functioning. So pay close attention every time I mention it. I will mention that it's the 57 nucleotide fragment. You can see that 57 nucleotide fragment in the band in the gel where the arrow is pointing. And you note that the thickness of the bands indicate the amount of product present. When pressed further, the Cas-A, Cas-B, and Cas-C knockout strains also contain this segment, but in higher quantities. They did not find this small section in Cas-D or Cas-E knockout strains. This led to the belief that this 57 nucleotide CRRNA may be cleaved from a pre-CRRNA or a precursor CRISPR RNA. When examining the impact on the delta case E strain, a long pre-CRRNA was found, perhaps indicating that when this gene is disrupted, pre-CRRNA cleavage is interrupted. The authors then proceeded to eliminate each subunit in turn until it becomes evident that without the Cas-E subunit, there is no small pre-CRRNA. They then wondered if that meant there was only the Cas-E subunit that was required to produce the small fragment through cleavage. The evidence seems to indicate that Cas-E is perhaps that essential unit. Moving on to the next experiment, the authors intended to use activity arrays with purified cascade. The results here in Figure 2c show that the complex is capable of cleaving E. coli K12 pre-CRRNA into that small 57 nucleotide fragment in vitro. But when the same treatment was given to another E. coli strain, known as E. coli UT189, there was no cleavage. The difference between the two is that UT189 has a different sequence, one that is not as recognized as the same by the cascade. This again helps us understand how CRISPR is truly phage specific. In addition, the RNA cleavage reaction proceeded without either ATP or divalent metal ions and then reached saturation within 5 minutes. Further investigation focused on if the Cas-E subunit alone was enough to instigate the cleavage activity on the pre-CRRNA. The authors explored this by creating a fusion of MAL-E, or maltose binding proteins, and the Cas-E subunit. Once more, as we can see in figure 2D, this fusion led, back again, to the 57 nucleotide fragment being produced without the complete cascade. This seems to support the author's supposition that Cas-E is the essential subunit for cleavage. However, they could not ignore the possibility that pre-CRRNA cleavage is an autocatalytic ribozyme-like reaction in which Cas-E is just an important RNA chaperone. In order to help them answer that question, the authors really dug further into Cas-E and found it utterly intriguing. It is a member of one of the largest and also the most diverse Cas protein families. When examining another Cas-E homologue, this time from a different bacteria, Thermophilus HB8, the authors found that the domain has two domains with a ferredoxin-like fold. This is very similar to some RNA-binding proteins. Mutating the 100% conserved histidine at position 20, which we are going to refer to as hist 20, into an alanine allowed the authors to examine the impact of this invariant histidine by removing it and replacing it with an inert alanine. In other words, the histidine that's located at position 20 was mutated, changed into an alanine, so that the protein structure, overall protein structure, really remained largely intact. But there was that change of that one protein in that position 20. The result was that even though the Cas-E subunit is still present, with largely the same structure, in its mutated state, it no longer produces our favorite 57 nucleotide sequence as shown in figure 2F to the right. Clearly, the authors concluded that the Cas-E subunit is essential for the cleavage ability, but that histidine in position 20 must be an important part of that ability as well. To continue their examination of the cleavage ability, and as you can see, the authors literally are following one clue to find the next answer, which then turns into the next clue. The authors are using a simultaneous expression of K12 type pre-CRRNA. And the result was fascinating. The authors found that by purifying the cascade, they were able to produce numerous copies of our 57 nucleotide fragment, which you can see in gel 3A there to the left. They then went on to clone and sequence these cascade-bound RNAs and found that 85% of them were CRRNA derived. Of those, 78% started with the repeat sequence AUAAACCG. With a closer view of 3B, we can see that the 5' end is well defined and followed by a complete spacer sequence. The 3' end is less well defined and ends in the next repeat section. Examination of the transcript for the single palindromic repeat reveals a structure that folds into a stable stem and loop configuration. The stem and loop consist of 7 base pairs, and the authors hypothesized that the structure helps with recognition by the RNA-binding Cas proteins. The pre-CRRNA cleavage site, or PCS, is found upstream of the 3' terminal end. The authors note that the 61 base pattern does not appear in the clone library. They suspect that this is an example in which at least 2 guanosines are removed from the cleavage process, which are taken from that stem-forming section. It is at this point in the article that the authors have reached their immune response segment. All of the previous tests and experiments have led them to this point in attempting to fully understand the CRISPR genes and their associated proteins. The authors examined the cRNA-loaded cascades and potential phage resistance. They created 2 artificial CRISPRs designed against phage lambda, each targeting 4 essential lambda genes. These coding CRISPRs produced cRNA complementary to both the mRNA coding and coding strand of the 4 genes. The template CRISPR targeted only the template strand of the same protospacer region. Finally, the non-targeting CRISPR with wild-type spacers served as the study control. The authors then used plaque assays with E. coli in order to determine sensitivity to the lambda phages. The results are shown by histogram in Figure 4a. The anti-lambda phage CRISPRs that only express cascade did not reduce sensitivity to of the host to the infection. The strains expressing cascade or Cas3 are much less sensitive to infection. But the template CRISPR made the host insensitive at even the highest infection titers. The coding CRISPR reduced sensitivity by 10 to the second fold, but of note, the phage resistance was lost when the cascade was omitted. Moving on to Figure 4b. The authors take a closer look at Cas3 and cascade Cas-E as the evidence points to them being an essential part of the immune response. In fact, strains containing Cas3 and cascade Cas-E H20A had a sensitive phenotype. This means that the pre-cRNA cleavage is mechanistically required for phage resistance. In addition, the co-expression of Cas1 and Cas2 really had no effect on sensitivity to the immune response. The authors suggest that these CRISPR-associated proteins must be involved in later stages, but not in this stage of the immune response. Overall, the key takeaway from this portion of the study indicates that the total sensitivity reduction is the result of the individual spacers working together in a synergistic manner, proving that independently they lack their full potential. In the discussion, there were 5 points the author conveyed in regards to their study. That first is that the complex of 5 Cas proteins is responsible for maturation of pre-cRNA to small cRNAs that are needed for an immune response. Secondly, the mature cRNAs have antiviral spacers surrounded by short RNA sequences from the repeat on either side of the termed 5' and 3' handle. These are likely conserved binding sites. Cascade-bound cRNA is a guide to direct the complex to the viral nucleic acid. Fourth, due to the anti-lambda CRISPRs of both polarities, reduce sensitivity to the phage. The cRNAs likely target virus DNA. And finally, the most important point the authors suggest is that transcription of CRISPR regions and cleavage of pre-cRNA to mature cRNA by Cas protein is the molecular immune response of prokaryotes that allows them to survive a phage attack. Again, with the benefit of hindsight, we can look at these conclusions and find them outdated, but the authors did an amazing job of following the clues provided in the lab and allowing the scientific study of CRISPR to evolve naturally to come to really at the time some groundbreaking conclusions. Overall impressions of this article, it really was kind of an interesting read. The authors took us on a step-by-step journey to better understand the role of CRISPR in bacterial immune response. I see this technology connecting us back to RNA world as another piece of evidence. The highly conserved nature of the nucleic acid, I keep thinking of especially those repeat sections that are so identical, and the importance of that histidine in the position 20. All of these things, I really see that highly conserved nature and of that nucleic acid and the adaptation of the genome to the invading nucleic acid points to just the incredible things that RNA is capable of accomplishing. As the first cellular life formed, archaea and later bacteria developed this immune response, likely in regards to early phages. However, if RNA is the first true life on Earth, then are we seeing a response in prokaryotes to that form of life or to the phages? It also makes me wonder about RNA as a potential endosymbiote, much like mitochondria and chloroplast. Again, this is coming from the concept of how the CRISPR became part of, how that protein became part of our genome and part of the prokaryotic genome, and how the virus portions are being absorbed into the genome and then later used as part of the CRISPR-Cas complex. It also, my apologies, was RNA absorbed into prokaryotic cells early on and then maintained as part of the cell because of some potential advantage? Is CRISPR and Cas an example of how it may have become incorporated? I don't know, but I suggest this based really on what the author's findings were. The ability of CRISPR to snip and incorporate portions of viral DNA into itself and then use that sequence to mount a defense is truly remarkable. Just as with the utilization of mitochondria in early prokaryotes, the utilization of CRISPR-Cas system that can incorporate and utilize pathogenic DNA is a really fascinating concept. Here is the first page of my works cited. These are about half of the works that I consulted in order to build this presentation. Obviously, it's going to include all of the assigned articles, but it also includes several other articles that I found while I was researching this particular work and the abilities of RNA and CRISPR specifically. I do appreciate your time and attention, and I hope you found this informative as well as easy to follow. The history of CRISPR is really the story of the ecology of prokaryotes during early life. Their utilization of RNA is an example of how these early life forms worked both together and against one another, and how this may potentially have driven evolution in the early life forms. Thank you again for your time and attention, and I really look forward to your critique of this work and this presentation, and I look forward to answering any questions that you might have.